No one have any questions? Thank you, ma'am. I'll start talking. So, okay. All right. It's just like the only tricky one is if it's a 5-membered ring. If it's 2 6-membered rings, then it's real obvious it's 1 4. If it's a 5-membered ring, then it can't form on this side because it doesn't have the OH. It has to form on this side. So it's a 1 2 linkage when it's a 5-membered ring. That's it.
Oh, I had a question. I think you said something wrong on one of those slides on accident, like backwards.
Oh.
I don't know if I was backwards. Yeah, it was like an alpha beta that you accidentally.
Oh, I know I said that backwards.
Oh, okay.
Yep. I was pointing to the right thing, and I said the wrong thing. Yep, I know for sure I said that backwards.
I forget which slide it was.
Yeah, no, I know when I turned it off, I was like, oh, you know what? I definitely said that backwards. I was wrong.
It was like oh no wait.
This one?
It's before that. Yeah, that one.
Yeah. Yeah, I know I had those backwards. Those are backwards.
Oh yeah.
That should be. Yep. Yep. Yeah, those are opposite sides. I knew as soon as I left that slide, no, like, as soon as I left that slide, I'm like, wait a minute. And there's no way to edit the videos. It's just you're stuck with what you recorded. So, yes, you are correct. I wrote them down backwards. Yeah, I said the right thing and I wrote down the wrong word.
A chakra is a lot like that.
It is. Yep. Alrighty. So my plan for today is to get us through at least the start of the metabolism chapter. Yep, perfect, thank you. I know, we're so excited to be back in class after a few days. So excited.
Did you eat candy last night?
A little bit. Peggy made a pretty good haul, considering she can't really walk. So, what's that? Oh, yeah, not really my thing. Yeah. Sorry. I know. Yeah. I guess I do 1 high ropes activity a year. My years are even. So, yeah, catch me again in. Not both beginnings. I did a marriage retreat at WindShape, and so 1 high element. Catch me again in January when my clock resets. And we'll see if I do another high element. Alright, so this is starting the last third of the semester where we look at metabolism. So this chapter is about intro to metabolism, then we'll look through glycolysis, then citric acid cycle, then oxidative phosphorylation, and the electron transport chain. And that's it. That's everything we have left for the semester. So really 4 chapters over 5-6 weeks. Awesome. It's like a chapter a week, and a lot of them are hopefully review in some kind of way. So citric acid cycle or glycolysis should mostly be, you probably have seen them in another class. So, and I will have the tests for sure graded by this afternoon. It's just I'm on that last question for everybody, and so you did not. You got like 6-7 hours left. All right. right, so metabolism is the overall process through which all living systems acquire and use free energy to carry out various functions. So we need to understand both chemical composition and three-dimensional structure to discuss metabolism. But chemical composition and the three-dimensional structure of glucose and all of its enzymes is not enough to tell us if the reaction is going to be spontaneous or non-spontaneous. So we also have to examine the free energy for metabolomic process, or metabolitic, how do I want to say that? Metabolism processes, metabolomics.
Can you just say metabolic?
Sure, that'd be great too. I can't spell that. Yes. All right. So in metabolism, there are two opposing reactions that can go on. Catabolism and anabolism. Okay, which one of these is the degradation process from all your biology courses? Yes. So catabolism is the degradation of free nutrients as well as cell constituents for the release of free energy. So then the opposite is going to be our anabolism. This is our biosynthetic pathway. That uses free energy to synthesize molecules. So then, if we're going to be talking about free energy, we've got to be talking about both exer and endogonic. Which one of those two is delta G less than 0? So exergonic is our delta G less than 0. Those are going to be spontaneous releases of free energy. Endergonic are non-spontaneous and require energy. So in our fancy little diagram here, macromolecules are broken down through catabolytic processes to release free energy, but then be used by anabolic processes to build back up macromolecules. So we're going to look at a little bit of both of these. So here is our little bit of biology for today. Just dipping our toe in some biology. There are a few different trophic strategies. They fall into two big categories. We got our autotrophs and our heterotrophs. Our autotrophs are organisms that are able to synthesize all their cellular constituents from simpler molecules. This could be a chemolithotroph, which is an organism. I can't spell that word. It's an organism that can synthesize all of its cell constituents from inorganic compounds. And photoautotrophs, as the name implies, uses photosynthesis to form all of its cell constituents. What we're going to be mainly concerned with is heterotrophs, which are organisms that obtain free energy through the oxidation of organic compounds. Such as glucose and lipids. So, how do we digest those glucose and lipid molecules? So digestion is going to be the intake of proteins, nucleic acids, polysaccharides, and triacylglycerols. That just requires the intake of all those things. So digestion reduces these polymers or macromolecules into their monomeric units. So proteins get broken down to amino acids. Nucleic acids are already their monomeric unit. I guess they could break down to nucleotides, that's possible. Into carbohydrates and fatty acids. Bless you. So, digestion takes place first starting in your mouth using the enzyme salivary amylase. So this enzyme works to break down your polysaccharides. Then as your food source travels down your intestinal system, your gastric and pancreatic proteases. Any guesses on what proteases break down? Proteins. So the proteins are broken down in the stomach and the pancreas. And then the lipases exist in the small intestine. And their job is to degrade triacylglycerols to fatty acids. So we've now broken down all of our macromolecules into monomeric units. So now we are ready to start sending them off to your cells for use. Your cells are going to take up these digestion products, these monomers, through the intestinal walls. It can happen in a few different ways. I guess. So your monosaccharides, your individual sugar units. So monosaccharides enter the cell through transport proteins. That is also true for amino acids. They have to be transported through the cell wall. Fatty acids, though, have a few different options. One option is that they can be transported into the cell. They can be modified back into a triacylglycerol. Or they can be modified with a protein for storage. So depending on what your fatty acid needs are at the time, you have a few different options of metabolic processes. So now that the cells contain these three different monomers, they could be stored or could be used immediately. We're going to talk about storage first. That's not really metabolism. So we're going to talk about storage, and then we'll spend the rest of the time talking about all the different ways that these monomers can be used in metabolism. So looking at monosaccharides, we'll do that first. So some will be used immediately depending on which monosaccharide you have. And what the energy needs of your body are. And the rest will be used to form glycogen, which is the storage molecule of sugar. Glycogen's a polymer of glucose, which is easy to break apart into individual glucose units. When you need energy, you can just cleave off a little bit. If there's glucose that's not being used to store as glycogen and not being used immediately, it can be converted to glycerol. What do we need glycerol for? To form our triacyl glycerols. So that glycerol is going to go off and turn 3 fatty acids into glycerol into triacyl glycerol for storage. I'm good on monosaccharides, things it can do. Use it, store it, convert it. Okay. Amino acids. Depending on what you need at the time, they can be used immediately to go off and form proteins. You have no mechanism to store excess amino acids. So if you've ingested more amino acids than your body has use for at the time, they will be converted into glucose and then stored as glycogen. So the end of the amino acid degradation pathway is if it can't be used at the time because you have no need for those particular amino acids, can then be converted to a glycogen molecule and they can be stored as a sugar unit. And we've already talked about lipids in the last, I guess, 2 chapters ago. Your fatty acids are just going to be stored in specific adipose tissue for use later. Yeah. Oh, sorry.
What keeps the amino acids from like continuing to just get built through?
It could be that where they are just doesn't need any more protein being built. So, or the particular amino acids that you ingested aren't the ones who are needed, or are not of the 20 essential, like you need some crazy amino acids, and your body's just like, well, I know I'll need sugar storage, so I'll go ahead and just store it later. So where do those sugars and proteins and amino acids go when they're needed? So what are our metabolic fuels? The metabolic fuels are all those small molecules we just looked at, amino acids, monosaccharides, and fatty acids. Those are all considered metabolic fuels because they can be used to build up larger molecules, or the breaking down of them releases free energy. So most tissues require energy from glycogen metabolism. So we build up our glycogen from either our monosaccharides or from excess amino acids. And the release of energy from breaking down glycogen is the favored method for most cells to get energy back. So glycogen looks like this. Right, so it's just a bunch of alpha-1,6 glucose molecules. So, in the presence of an inorganic phosphate molecule, that should be O. So we have our inorganic phosphate. And glycogen phosphorylase, which, as the name implies, phosphorylates the glycogen. It will break off a single glucose I'm not great at drawing six-membered rings, apparently. So it breaks off a single glucose, combines it with the inorganic phosphate to make glucose 6-phosphate. The rest of your glycogen is still, I'm not going to redraw that, but still, see over here, just short one carbon, or one glucose now. And this now is ready to enter glycolysis. Any questions on that? Pretty straightforward. So this is your sugar storage, your glycogen storage can undergo the release of a single glucose to enter glycolysis, which is how most of the glucose enters your glycolysis stream. We can also mobilize some other ones. So that is our monosaccharides. We can mobilize your adipose tissue. So adipose tissue mobilization only happens when you're low on, your body's going to choose free glucose first, then it's going to choose glycogen second. So you're only going to be able to get those molecules out of the adipose tissue if it's used up its reserve of these first two options, which you know is the basis behind every single exercise plan out there. So adipose tissue mobilization happens through your triacylglycerols being hydrolyzed. So your triacylglycerol molecules are hydrolyzed by the enzyme lipase into free fatty acids. So fun fact, your heart tissue predominantly uses free fatty acids as its source of energy, which is why everyone makes a big deal about eating your healthy fish, omega-3 fatty acids. That is the preferred energy source for your heart muscle. But that's really the only body mechanism that uses free fatty acids. Like if you ate a ton of omega-3s, it's just going to be stored as regular fatty acids. Right, your heart's going to use as much as it needs, and then it's going to put everything else into storage. So eating a high-fat diet does not necessarily mean, well, you could have a fairly healthy heart, but you're going to be on the heavy storage side.
There's too much fat around your heart.
Yeah. Right, and that's one place it can store it. It can store it around your heart. It can store it like in the little vessels. So you could be on the too much storage end of it. So that's my other notes. I'm not going to say convert, it can be used. So in a mechanism that we're not going to look at this semester, but if you take Biochem 2, they spend some time on fatty acid metabolism and catabolism. Fatty acids can enter a cycle where ATP production is one of the outputs of the cycle. So that's another option. And then finally, amino acid mobilization. So the obvious one is that amino acids are used to build proteins. They're also used for the degradation of proteins that are either not being used or have been formed incorrectly. So they're used in that process. And if they're not being used, like I said earlier, they can be stored. They can be converted to stored as glycogen if there's no current need for them. Get all the mobilization options. All right, so you may be thinking it's a lot of different things, and I'm not exactly sure where we're going with all this. Well, besides the citric acid cycle and glycolysis, all of these cycles actually interrelate. So many of the steps required to break down your monomeric compounds or to build up to polymeric compounds share common intermediates along their metabolic pathway. So several molecules appear either as precursors or products in many pathways. We call those common intermediates. Not common as in they're found everywhere, but common as in they're shared intermediates. So for instance, in glycolysis, which is impossible to see, my little tiny figure down here. Oh, okay, there we go. Still not great. Okay, glucose is degraded to glyceraldehyde 3-phosphates, to pyruvate, to acetyl-CoA, which I have slightly bigger over here. All three of these intermediates appear in other pathways. So pyruvate is, oh, here we go. Pyruvate is needed for the citric acid cycle. It's needed for glycolysis. It's needed for fatty acid synthesis. It is also needed for amino acid synthesis, just for starters. Citric acid cycle. Acetyl-CoA is needed for mostly citric acid cycle and glycolysis. but you'll see it in a few other pathways. Oh, fatty acid synthesis, that's the other one.
For pyruvate, are you saying that it's involved in the Giovanni cycle? Those four things are all three of different involved?
Just pyruvate does all four of these. Acetyl-CoA does these three. Glyceraldehyde 3-phosphate is needed for glycolysis, photosynthesis, and fatty acid synthesis. That is a compound that multiple pathways need to have it made in order to do all those different metabolic, I don't know why I'm having a hard time with that today, metabolic processes. All right, there are going to be 3 molecules that are super important to know, and you will need to be able to identify these. I'm not going to have you draw them, but be able to identify them. If I can get them all in here at once. NAD, FAD, and CoA. We will draw all the mechanisms and all the steps for all the other cycles. So I figure this should be a gimme, just if you recognize these guys. I heard somebody asking a question. What? Yes, this one's FAD. Yep. Yep.
So we just need to be able to recognize.
Just these 3 just to be able to recognize. All right, let's see if I can zoom in a little bit. We might not get to all of these, but okay. So this one's NAD. Which stands for nicotinamide adenine dinucleotide. And its job is to carry electrons to and from reactions. It's kind of hard to see, but it's technically NAD+. The little plus is right here. Plus. On that nitrogen, there's a plus sign. NAD+. So in this one right here, that nitrogen gets a, let's see if I can do that. This nitrogen is currently positively charged. When it becomes, when we add the extra proton, which we'll look at, I guess, Monday, to convert from NAD+ to NADH, it loses that positive charge. The reaction is NAD+ plus a proton plus 2 electrons makes NADH. So its job is going to be transporting electrons, in this case taking them, or in the reverse case donating them. So it's reversible. All right, we'll stop there. We'll do FAD, CoA, and our discussion in depth about thermodynamics on Monday.